concentrations of specific proteins define and specify body regions in the embryo. And we've talked about several types and names of maternal genes and their role in anterior and posterior patterning. So yeah, we've talked about our favorite gene, bicoid, and other maternal genes such as nanos, hunchback, and caudal. They're all morphogens. So these morphogens can be detected as gradients of proteins, and they can be visualized by immunohistochemistry. And in addition to these morphogen proteins, we've talked about other proteins that are not morphogens, that are maternal genes that are important for body patterning in the early embryo. And so another example of this is the torso and trunk genes that are part of this receptor tyrosine kinase pathway. And they're important for defining the terminal ends of the embryo. Okay, and then we've also talked about maternal genes that are involved in dorsal ventral patterning. And so we talked about this signaling pathway that starts with toll receptor, and it ends up in a signaling cascade that activates this protein called dorsal, which is a transcription factor that needs to be translocated from the cytoplasm into the nucleus to direct those cells that have the dorsal protein in the nucleus towards the ventral fate. Okay. Okay, and we talked about how this dorsal ventral patterning and the specification is required for normal gastrulation and for the beginnings of forming the different germ layers, the ectoderm, the endoderm, and the mesoderm. Okay, so the maternal genes get activated upon fertilization, and then they activate genes in the zygote, so zygotic genes. And so we talked about how dorsal regulates expression of the zygotic genes DPP, twist, and SOG in that kind of gradient model that we talked about, where DPP activity gets concentrated on the dorsal side of the embryo, and twist gets expressed on the ventralmost cells. And then we started talking about gap genes in anterior and posterior patterning. And so some of these gap genes include zygotic hunchback, which also acts as a morphogen and acts like a morphogen as you would expect. There's different concentrations, and they control the expression of other gap genes in specific locations. Okay, and so some of the experimental techniques that we've just talked about are in situ hybridization, immunohistochemistry, and reporter genes. So I just wanted to make sure, I thought after the last lecture that these techniques were not coming across as clear, so I wanted to kind of review this with you. I did. I posted these gene expression PowerPoint slides under supplementary notes on top. Okay, so, and I apologize, I got this off the internet, and I can't find the website that I got it off, so I have no citation, which is bad. This is not mine, okay? But we're not going to distribute it. I'm telling you right now it's not mine, so it's not plagiarism, but it's a very good, good set of slides, okay, for you to reference. Okay, so when you study gene expression in the embryo, there's different ways that we can look at it. There's in situ hybridization, which probes for mRNA. There are probes for protein, which is immunohistochemistry. And there's another tool called reporter genes. So mRNA, when you look for mRNA or visualize mRNA on a gel, that's called a northern. And when you visualize protein on a gel by size, that's called a western. So if you were to think of in situ hybridization as kind of a northern and immunohistochemistry as a western, the only difference is what you're probing for, mRNA or protein. And reporter genes allow you to use immunohistochemistry for the most part when you don't have a specific antibody for your protein of choice. Okay, so most of the methods can be used in tissue sections or whole mounts. Okay, so for in situ hybridization, you prepare your fixed tissue and permeabilize the whole mount. And then you have a probe for your mRNA that you want to locate. And the probe consists of what's called antisense RNA or antisense DNA. So basically, this is just a sequence of nucleic acid that complements the sequence of the mRNA. And the probe can be either fluorescent or radioactive, or can be attached to, detected using an enzyme. So you can visualize the location of your mRNA by using the microscopes. Okay, so this is the slide that I showed you in section today. So on the left, it's showing you different ways of detecting mRNAs. So here, the mRNA of interest is in red. So let's say it's bicoid, okay? And the probe is shown here in black. Okay, so this is a DNA sequence that's complementary to this mRNA sequence. And the probe can either be fluorescently labeled, it can be labeled with a metal such as silver, or it can be basically physically attached to a protein called, well, to a protein to which an antibody can bind that's linked to an enzyme, and then a substrate can be visualized. So it's a substrate, it gets cleaved by this enzyme to produce a product that you can see. Okay, so this is similar to what we talked about for immunohistochemistry during section, where the only difference is that instead of using an mRNA as a probe, you're using a primary antibody as a probe to bind to a specific protein, and they can be fluorescently labeled or labeled with a metal or enzyme linked. Okay, so if we go to our slides from last lecture, okay, on the top is in situ hybridization of bicoid mRNA. So you can see the mRNA is very restricted to the anterior portion. And then immunohistochemistry, or using antibody staining for bicoid protein, is shown below it. So there's different patterns, right, when you compare the in situ hybridization and the protein levels. And this is what's shown here. Okay. Yeah? Okay. So I also introduced in the last lecture this tool called reporter genes. And so a reporter gene is basically a sequence of DNA that contains regulatory regions that transcription factors can bind to. And you hook those sequences to something that you can visualize later. So in this case, it's the gene that encodes beta-galactosidase, or called LAC-Z. So you make this DNA construct, and you inject it into a zygote. So you introduce it into an embryo. And wherever the transcription factors are present that can activate or bind to this regulatory region, you'll see expression of the LAC-Z protein. And so you can detect where LAC-Z is located using immunohistochemistry. Okay, so you have beta-galactosidase, which is encoded by the LAC-Z gene, and you can add to your section this substrate called X-gal. And when X-gal is present and beta-galactosidase is active, you get basically a color. And so you can use immunohistochemistry to detect it. Another reporter that is used often is green fluorescence protein, or GFP. So instead of LAC-Z, you would put in the GFP gene. And then you can just use fluorescent microscopy to see where